Greetings, everyone. Hello and welcome. I would like to introduce myself. My name is Riham Adel Abdel Hafiz. I'm clinical pharmacist. I have master degree of clinical pharmacy, Helwan University, PG certificate of clinical pharmacy, Queen's University from United Kingdom, diploma degree of hospital pharmacy, Cairo University, Egypt, diploma degree of cosmetics manufacturing, Cairo University, Egypt, bachelor degree of pharmacy and drug manufacturing, Must University, Egypt. And right now, my working as clinical pharmacist in hospital. So let's start our journal club today. Today, I'm going to present a published paper that published at the New England Journal Medicine at 2018. It's titled about balanced crystalloid versus saline in critically ill adults. So let's start. Introduction. The study trial was used to determine the effect of isotonic crystalloid composition on clinical outcomes in critically ill adults. They conducted the isotonic solution and measure adverse renal event trial SMART, SMART, which compared the use of balanced crystalloids with the use of saline in patients in medical and non-medical Smart Med and Smart Surge intensive care units. They hypothesized that the use of balanced crystalloids would result in a lower overall incidence of death, new renal replacement therapy, and persistent renal function dysfunction than some methods. They conducted a pragmatic, unblinded, cluster randomized, and multiple crossover trial, in which the use of balanced crystalloids was compared with saline for intravenous fluid administration among critically ill adults who admitted to 5 ICUs at Vanderbilt University Medical Center between June 1, 2015 till April 30, 2017. The trial sites and patient population, the patients had an age 18 years of age and above, who were admitted to a participating ICU during the trial period, were enrolled at the ICU admissions, and enrolled patients who were discharged from the hospital were eligible to participate again if they were readmitted to participating ICU. Randomization. For each month of the trial, participating ICUs were assigned to use either balanced crystalloids or saline. For any intravenous administration of isotonic crystalloid, ICUs were randomly assigned to use saline in even numbered months and use balanced crystalloid in odd numbered months, or vice versa. Treatment. Patients in the saline group received 0.9 percentage sodium chloride when intravenous isotonic crystalloid was administered, whereas patients in the balanced crystalloid they were received either lactated Ringer solutions or plasma light A, according to the preference of the treating clinician. An electronic advisor within the electronic order entry system informed providers about the trial, asked about relative contraindications to the use of balanced crystalloids as hyperkalemia and brain injury. The treating clinician determined the severity of hyperkalemia or brain injury at which saline used rather than using balanced crystalloids. The unassigned crystalloid was also available from the pharmacy when clinicians believed it to be required for the safe treatment of any patient. Data collection. The data collected as information on pre-enrollment renal function, demographic characteristics, diagnosis, predicted risk of in-hospital death, etc. First outcome, or we can say it primary outcome, was the proportion of patients who met one or more criteria for a major adverse kidney event within 30 days, the composite of death, new reset of renal replacement therapy, or persistent renal dysfunction. Patients who had received renal replacement therapy before enrollment were ineligible to meet the criteria for new renal replacement therapy or persistent renal dysfunction, but could qualify for the primary outcome if they died in the hospital. Secondary clinical outcome included in hospital death before ICU discharge or at 30 days or 60 days, as well as ICU free days, ventilator free days, vasopressor free days, etc. Secondary renal outcome included new receipt of renal replacement therapy, persistent renal dysfunction, acute kidney injury of stage 2 or higher. Statistical analysis types. They planned to retain adequate power to detect the targeted difference, so they raised the sample size from 8,000 to 14,000 patients, and they extend the trial period from 60 to 82 unit months. The Data and Safety Monitoring Board conducted two interim analyses. The primary analysis compared the incidence of the primary outcome in the balanced crystalloids and saline groups with a generalized linear mixed effects as fixed effects and random effects, fixed effects as age raised 6, and random effects as ICU where the patients admitted. The secondary analysis, they compared secondary outcomes between the trial groups. They performed subgroup analysis according to type of ICU, source of admission, receipt of mechanical ventilation, receipt of vasopressor, etc. They conducted sensitivity analysis using alternative approaches to addressing the issue of missing data on baseline creatinine level. Then they performed sensitivity analysis according to the volume of crystalloid administered, accounting for crossover and limiting the analysis to each patient's first ICU admission. Other between group comparisons were made with the Mann-Whitney rank sum test for continuous variables and the chi-square test for categorical variables. Results. The median age of the trial was 58 years old, and there was 57.6% of the trial was men, and greater than one-third of the patients has received mechanical ventilation, one quarter of the patients has received vasopressor. There is no significant difference in the baseline characteristics between the patients assigned to receive balanced crystalloid and those assigned to receive saline. The median volume of balanced crystalloids that administered to balanced crystalloid groups was 1000 milli, and the median volume of 0.9% sodium chloride that administered to the saline group was 1020 milli. There was no significant difference between groups in the median volume of non-isotonic intravenous fluid, blood products, or medications administered. Fewer patients of the balanced crystalloids rather than saline group has plasma chloride concentration greater than 110 millimole per liter, 24.5% versus 35.6%, or plasma bicarbonate concentration less than 20 millimole per liter, 35.2% versus 42.1%. Differences between groups in chloride and bicarbonate concentration were greater for patients who received larger volume of isotonic crystalloid. A total number of patients 1,139, 14.3 percentage of balanced crystalloid group, and 1,211 patients 15.4 percentage of saline group had major adverse kidney event within 30 days. In pre-specified subgroup analysis, the difference in the rate of the primary outcome between the balanced crystalloid group and the saline group was greater among patients who received larger volume of isotonic crystalloid and among patients with sepsis. A total number of patients, 818 patients, 10.3 percentage of the balanced crystalloid group that has died in the hospital before discharge within 30 days of ICU admissions, compared to 875 patients, 11.1 percentage of the saline group. A total number of patients, 189 of the balanced crystalloid group and 220 patients of the saline group had received new renal replacement therapy. The highest stage of acute kidney injury and the incidence of persistent renal dysfunction did not differ significantly between groups. Discussion. In preclinical models, high chloride content of saline have causes hyperchloremia, inflammation, acidosis, hypotension, acute kidney injury, and death. Healthy volunteers have suggested that saline may decrease renal perfusion due to chloride-mediated renal vasoconstriction. Critically ill adults shows higher rates of acute kidney injury, renal replacement therapy, and death with saline rather than using balanced crystalloids. The use of balanced crystalloids rather than using saline have shown absolute difference of 1.1 percentage points in favor of balanced crystalloids in the primary outcome. Conclusion. The results suggest that the use of balanced crystalloids rather than saline might prevent one patient among every 94 patients admitted to an ICU from the need for new renal replacement therapy, from persistent renal dysfunction, or from death. Moreover, the difference in outcomes between balanced crystalloids and saline appeared to be greater for patients with sepsis and patients who received larger volume of isotonic crystalloid. The appropriate composition of a fluid may depend on the indication for its use and the condition of each individual patient. Concern that the relative hypotonicity of balanced crystalloids could increase intracranial pressure in patients with brain injury. So that led us systematically to use saline rather than using balanced crystalloid for patients who have brain injury. Trial strengths and trials limitations. So let's start by the strengths first. The large sample size provided statistical power to detect small differences in patient outcomes. The trial design allowed delivery of the assigned crystalloid early in each patient's critical illness. Enrolling all adults admitted to participating ICUs and allowing clinical providers to deliver the assigned crystalloid during clinical care minimized selection bias, and improved generalizability. Trials limitations conduct at a single academic center limits generalizability. Clinicians' decision to initiate renal replacement therapy may be susceptible to treatment bias. Censoring data collection at hospital discharge may underestimate the true incidence of death at 30 days and may overestimate the true incidence of persistent renal dysfunction at 30 days. On the basis of the hypothesized mechanism of chloride-induced organ injury or acidosis, so this trial doesn't inform the choice between lactated ringer solution and plasma light aid together. Thank you so much for watching my video and I hope you enjoy it. See you soon. Bye.